All right, well, we were waiting for you, Alex.
Oh, yeah.
Yeah. Okay, good. Okay, that's working. Okay. All right, so let's start. Let's have a little chat about chapter 9. All right. So today, sort of as promised, we're in chapter 9, part 2, which is going to be transcription and translation at minimum, and hopefully we'll get a little bit of mutations in, or gene transfer, whichever. So last time we talked about DNA replication, how all the DNA gets copied. This time, it's important to remember that in replication, all the DNA is getting copied. The pieces we use, the pieces we don't use. In bacteria, it's almost all used by all the DNA. Transcription, which is the next process that we're going to talk about. The cell is going to copy only one of the DNA strands. Only one. And it's not just going to copy it as DNA, it's going to change it into a different nucleic acid. So I'm going to say it's copy into messenger RNA. That's why we call it transcription and not replication again, because we're transcribing. It's like transcribing something from another language, right? So you transcribe it into your own language. And this RNA language, that nucleic acid is just slightly different, but it's a different enough, okay? So I want to digress for a minute and talk about the different types of RNA and what's different about RNA from DNA. And then we'll go through the steps of transcription, only 3. Initiation, elongation, termination. It's much shorter than replication, okay? Not as many enzymes at all, easy peasy. So RNA stands for what? Ribonucleic acid, good. Right? Anybody remember what's different about it? It does. Yes, there are no T's in RNA. Right? We'll put the sign with the line through it. No T's in RNA. T is replaced by U for uracil. So when DNA has an A in it, the complement in RNA is not going to be a T, but a U. Right? When the DNA has a G, the RNA is still going to code a C. When the DNA has a C, the RNA will still code G. And when the DNA has a T, complementary of the T is still going to be A. Okay? Sometimes students forget and they want to put U in twice. That's why I go through that. Ribo instead of deoxyribo. So that means that it has ribose as the sugar. The rest of it's the same. Still is phosphates. Uracil is replacing T. T is a pyrimidine. Uracil is a pyrimidine. Same. So purines, pyrimidines. Most RNA, but not all. Most RNA is single-stranded. Okay, if I do that, SS for single-stranded rather than write that out. Okay. Different types. So the type we're going to talk about the most is messenger RNA, at little m, right? Messenger RNA. And it is single-stranded. And the pieces of it are quite long. I would say because the codons, which are sets of 3 RNA bases, 3 codons to 1 amino acid, and most proteins or protein subunits are in the neighborhood of a couple hundred amino acids, so times 3. So messenger RNAs, like the longest ones, are probably like 600 bases ish. Okay. On the longer end, let's say longest would be about 600 bases. So when I draw a messenger RNA on the board, I'm not going to draw 600 bases, but I want you to try to remember it's long and single-stranded. Do you remember the other kinds of RNA? tRNA, which stands for transfer RNA. That's okay. Nobody can remember that. Nobody can remember that one. Transfer RNA, right? Transfer RNA doesn't like to be single-stranded. It's the one that forms that weirdo like cloverleaf shape, right? So I would draw it kind of like this. There's a little hook there and a loop, and then another loop, and then another loop, and then a tail. And there are places where it's double-stranded and places where there's these weird loops, right? There are 20 different transfer RNAs that have different anticodons down here at the bottom. One for each of the 20 amino acids. Actually, there's more than that, but let's just say 20. This obviously is going to be used in the ribosome with the messenger RNA to equal protein. Let's just do it that way, right? To equal protein, chains of amino acids. The third type of RNA that we're going to just briefly mention is ribosomal RNA, which we designate little r RNA like that, because it's part of the ribosome. It's important. It's short, double-stranded, usually in 3 pieces. Okay. It's different densities, which relates to size and length, right? Size and how big it is and the shape that it's maintaining. In eukaryotes, prokaryotes, and archaea. That ribosomal RNA is what gave us the 3 domains. That's what's the most different between the 3 different types of cells, is what kind of ribosomal RNA they have. So basically how they make their proteins, which since proteins do everything, you know, blah, 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 blah. You know the rest of that story. These are just 3 types of RNA. In bacterial cells, there are about 8 that we know of, or 10. In your cells, there's about 12 to 18 different types of RNA. Most of them function as regulation, meaning they help genes either be transcribed and translated, or they suppress genes from being transcribed and translated. Right? So this is just a tiny glimpse into the world of molecular biology, of how genes are actually regulated. For us, that's enough. We're just getting started, right? All right, so having said that, let's talk about the transcription process that we're going to talk about. The cell does that for all 3 kinds of RNA, but we're just going to focus on the making of the messenger RNA. Right? Because that's when you open up a bacterial cell and you look and you ask how much RNA is here, most of it is messenger RNA. So that's why we're going to focus on that one. But the same process happens to make message, transfer, and ribosomal RNA. All right. So, like I said, I don't draw that picture. So I'm just going to write the words. And then we'll look at the picture. Okay, so transcription. The steps are at first initiation. And that is going to involve a specific sequence of DNA that we call the promoter. So sequence of DNA. The most common promoter is called the ta-ta box because the sequence is TATAAA, right? And then, obviously the complementary. It's found at the beginning of most bacterial genes. It's how the cell knows this is the place to start to do transcription. The enzyme that recognizes the promoter, it's called RNA polymerase. Come to genetics, learn more about how that actually happens. It's way more complicated than this. We're doing the Goldilocks and Three Bears version, right? Simple, simple. RNA polymerase recognizes the promoter. RNA polymerase only is going to copy one strand of the DNA. It's going to copy the 3' to 5' strand, which we call the template strand. And it's going to make messenger RNA 5' to 3', just like DNA polymerase did. It's going to add bases, all right? Not quite as quickly as DNA polymerase, but it's going to add bases or nucleotides. Its proofreading capacity is not as good as DNA polymerase, but there is some. We used to say RNA polymerase does not proofread, but now the research tells us it does a little bit. So I'll just write weak proofreading. How about that? And why that's important is because it's much more likely that RNA polymerase is going to make a mistake and leave it than DNA polymerase is going to make a mistake and leave it. And when you think about maintaining the continuity of all your genes, you want DNA polymerase to be better at fixing the mistakes than RNA polymerase, right? RNA polymerase might make a mistake that might change a protein a little bit, but it might not affect your ability to function. The second step is elongation, and that's actually just, I'm going to write transcription bubble. Oh, give me a minute, I'll remember. Maybe 8 to 12 bases of DNA are undone at a time. Don't write that down. I'm not sure that's true. But a small section of DNA is going to be opened up, and we're just going to copy a little piece of it at a time into messenger RNA. And then termination. That's rather self-explanatory, right? Gene over, stop. And like the promoter, it's a particular sequence. We actually call it the terminator. At the end of the gene, that when RNA polymerase gets there, you can just think of it as stopping or detaching. Again, come to genetics, learn more about it. So let's look at some pictures. It's okay if I pull this down. You good? Yeah, get that perfect. Yeah, so let's look at some pictures. This initiation, right? So the double-stranded DNA, they're making one strand red, one strand blue, right? So they're calling the red strand the coding strand because that's going to have basically the same sequence of bases, just the T's are going to be replaced with U's. The 3' to 5' strand is the template strand because that's the strand the RNA polymerase is actually going to use to copy, because remember, it works 5' to 3'. So RNA polymerase is going to recognize the promoter, which they haven't depicted out for you, which is fine. It's going to bind here, and the RNA polymerase is kind of this orangish yellow enzyme. And we don't need helicase, we don't need gyrase, because we're just opening up a little bit. Like I said, I think it's about 12 So 12 or 25, I forget. We open up with just a little piece of the DNA and the RNA polymerase is going to copy. And then as soon as the RNA polymerase is here, this is going to rewind and the next little piece is going to be opened up. So we don't need all that elaborate helicase, gyrase, single-stranded binding proteins, primer, all that nonsense. Because RNA polymerase, like primase, I don't know if I gave you guys this analogy. I gave you the railroad track analogy. Yeah. So primase, like primase, RNA polymerase looks at the rails on the railroad track and the ties of the bases and is like, okay, the promoter is here, is where I start, and that's the 3' side. So I'm going to start adding 5' bases complementary to that. So, and you can see the bases being, okay, this is just showing the promoter there. Let's see if we can get one that actually shows. Really? That's all you're giving me? That's kind of lame. Okay, so this is elongation. Sorry. So this is showing you actually the RNA polymerase is moved, right? Before it was here, now it's here. And all the yellow are the bases from the nucleotide pool that are RNA bases that have been added, right? 5' to 3'. So like I said, then this little area is going to close and this message is going to hang out like down this way. And the next area is going to open up and the polymerase is going to chug along. And then it's going to keep going until it gets to the red spot. What do you think that means? That's the terminator. Yes. Right. Then when RNA polymerase gets here, then gene over. Okay. Trying to think if there's anything else I need to tell you about transcription before we move on to translation. I don't think so. I think that's about all you need to know. 5' to 3', less proofreading, needs a promoter, needs a terminator, elongation. All right, I'm just going to write under elongation the transcription bubble. I'm going to put small area of DNA that is unwound and unzipped to use as template to add RNA nucleotides, things like that. Okay. I'm going to leave replication here and transcription, and I'm going to write translation underneath it to kind of remind you sort of the phases of how this works. And I would remind you that all the different types of RNA are all made the same. So there are genes to transcribe for all those transfer RNAs. There are genes that gets transcribed to make ribosomal RNA that goes into the ribosome. Okay. Translation. Only messenger RNA gets translated, not the other types. So translation is when we take a messenger RNA, and your book says that most genes on average are about 400 amino acids long, I guess, because it said that the messenger RNA can be as long as 1,200 nucleotides. I think more of them in the neighborhood of 600 to 900, but let's say 600 to 1,200 nucleotides. And in order to translate or convert the message in the messenger RNA into amino acids, which get strung together to make peptides, which then get secondary, tertiary, quaternary structure to make them into proteins, like functional proteins, right? In order to do this first part, get the messenger RNA, which is basically a copy of a gene from the DNA, into amino acids. We need more equipment than we've talked about so far, right? We need a place for this to happen. Place, a site, a location, which you all very successfully told me when we were doing chapter 4, that's the ribosome, right? Because I asked you, what does the ribosome do? And you all said protein synthesis. Again, I would remind you the ribosome has 2 parts, large subunit, small subunit. We need transfer RNA that has amino acids attached to it. So we say that's charged transfer RNA. With an amino acid and amino acid. And then, of course, we're going to need the messenger RNA itself, right? That the RNA polymerase just made. Just like replication and transcription, translation happens 5' to 3'. So the 5' end of that message is what is going to fit into the ribosome in order for that to get started. As before, I'm going to write some words and draw some pictures. Amino acid, sorry. No, thanks for mentioning that. 7 amino acid. There we go. So, right, the general purpose is to actually make the proteins the cell can use to do work, like enzymes in the Krebs cycle, right? The enzyme, the proteins in the electron transport chain. The how the cell does this in the ribosome. That's what we're going to talk about next. Process of translation. So translation, like to change into another language, right? So translation, we are changing nucleic acid language into amino acid language, and we need a dictionary for that. We need something that tells us what the words are in the other language. Okay, that happens to be what we call the amino acid chart. Right, it only translates messenger RNA. So it's only messenger RNA codons. Again, come to genetics, learn more about how we know that the codons are in sets of 3, but they just are. For you, you just have to know that. So a codon is a set of 3 nucleotides, RNA nucleotides. That is, I'm going to put in quotes, that is a word in RNA language that we are trying to translate into amino acid language, protein language. Right, just like you know the word for table in Spanish and the word for table in English. Yeah, well, you have to know the word in both languages, right? If you don't know the word in one language, you have to have a dictionary. I don't know one has those anymore. Okay, you have to have the app on your phone that you can just type the word in or record the person what they're saying to you, and then the app tells you what they said, right? Somebody has to do the translating. So the chart, the infamous chart. Let's see if I can find that. There it is. But wait, how do I do that again? All right, go back here. Yeah, really, I need a whole semester of training to be able to do that. Trial and error. It took me like, what, 2 seconds, and I'm slow with this. Okay, so here's the chart. You have to memorize it for the test. Just kidding. See if you're awake. No, I will give it to you for the test. No one has to memorize this ever. Right? That's like memorizing all 20 amino acids. What's the point? It's always in a book or on Google, and that Google can get right. Right. So same with the amino acid chart. I will provide it to you on the test day. It might not look exactly like this one because I think the one I copied is from your BIO1 book, but they're always the same. Okay. Every one of them is set up exactly the same. Over here is the first base in the triplet or in the codon. That's why it says first base position. And they're always in this order, U, C, A, G. Because a long time ago, like 1957, I think, they decided that's how the chart was going to be put together. And that's how it's put together every time. U, C, A, G, U, C, A, G. So first position, second position, third position, letters in the word, right? In RNA language. And look, it's a dictionary. It only has one page. It's a dictionary. It tells you what amino acid those letters go with so that you can translate the language. So most proteins, most messenger RNA transcripts begin with AUG. Most proteins don't keep the AUG, but that's a different story for another time. Most of them do begin with AUG, which is methionine, which sometimes when it's at the beginning of the protein gets formulated, which is why there's an F on it, a formal group gets added to it. That's why it's labeled as start, because when the scientists were first doing this, they found out that almost all the transcripts, the messenger RNA they got out of cells started with AUG. So they labeled it start, right? The other 20 amino acids, I hope you at least recognize their names, right? Proline, threonine, alanine, glutamic acid. I do want you to take a quick look at this and notice a couple of things. You see how proline here, CC anything codes for proline. Proline is an important amino acid because it causes a bend in the primary amino acid chain, which is going to help that primary chain of amino acids, that primary polypeptide, to form alpha helix or beta pleated sheet. So prolines are structurally important. So it's kind of important that if you're supposed to get a proline, you get one no matter what, basically, right? CCU, CCC, CCA, CCG, they all code for proline. Some of the other ones, notice leucine has two different codons, UUA, UUG, and then all of these CU, whatever. So some of them, like leucine, they have 6 transfer RNAs that are specific for just leucine, right? Others, like serine, only 2, and this is the only code for methionine, AUG, right? So that one's unique. See the red boxes? Right? We've been taught for a long time, red means stop, right? Green means go. These 3 codons, UAA, UAG, UGA, there are no amino acids for them. They are at the end of a long chain in messenger RNA, and they mean stop, protein over. Everybody disassemble, right? And those are universal. Those are the same in you as they are in bacteria. All of these are the same. Again, I will give you the chart, right? You might have to tell me what the template or the coding strand of DNA is that went with the message I gave you. You may have to take the message and tell me what is the order of the amino acids in that message. And after we talk about mutations, I may ask you if this particular mutation happened, what will happen to the protein? All right, so you've used the chart before, yes. Sort of. Oh, yes. Cut me deep. But, so I will, after we talk about translation, let me erase this. And I'll write out an amino acid chain that we can use when we're doing our translation practice here. So I'm gonna erase all this nonsense. And I'm actually going to turn that off for a second. Okay, so this is going to be your messenger RNA, 5 prime, AUG, C, we'll make a proline, CCU, AGA, CUC. AAA, AGC, I like that one. Let's put CCC on there and GAC, and that's good. Let's do UGA. So I'm just going to project the chart over the top of that rather than pull the screen down so that you can see most of it to do it. Take a minute. Tell me if you can figure out what the order of the amino acids would be in this particular string of messenger RNA. It's kind of worth doing. Let's use purple. I guess I should have written it a little bit higher. I don't think it'll give you much trouble. No. Oh, sorry. Well, if I had that, $7,000 training, that probably wouldn't have happened. You done? Yes. That's not enough time? Okay. Okay.
I know. I've been happy to know you want, but
Okay, did everybody do some of it? Let's start with that. Yes? Okay. Yeah, except Devin, I know, he's back there playing video games. All right, AUG. I'm just teasing you. So we already know that one, right? So first position, A, I usually do this. I usually put a line under, or more likely I do this. Every 3, I put a line, so because I'm old, C's and G's look alike, right? And so I don't want to make a mistake of substituting one for the other while I'm trying to figure it out. So I usually do this. I usually just put a horizontal line like that. I mean, a vertical line like that. Okay, so AUG, first base, A, second base, U, and then I come to this box and I look for AUG, which is what? Methionine. Good. CCU. That's proline, yeah. AGA. I don't know that one. A, G, so AG, A, yep, arginine. Good eye. Arg, CUC. That's leucine, yep, LEU, yep. Triple A. So first position A, second position A, I come to this box, and what do I find in here? Lysine, LYS, good. AGC, I don't know that one. Serine, they say. AG, this box, AGC, serine. Yep, okay, I'll agree with you. CCC, that's got to be proline again. Oh, very bendy amino acid chain we've made. GAC, aspartic acid. I think that one's ASP. and UGA. Stop. On the test, I may forget to make a stop. I promise there will be a start program, but I can't promise there'll be a stop, right? Because sometimes I'm just typing away and then whatever. And remember that on a test, if I give you even a sequence this long, that's barely a peptide, right? So I'd be giving you just a tiny little piece of the messenger RNA chain that you'd be trying to translate. You guys could do that, yeah? Okay with that? Comfortable with that? I'll give you the chart. You can do that, right? Lovely. Likewise, if I asked you the complementary DNA strand from this, right? The 3' strand, you could do that. What would be complementary to A in DNA? T, and what would be complementary to U in DNA? A and G, C, yeah, blah, blah, blah. You could do that, right? Okay, lovely. Okay, so now let us then talk about how translation takes place. So that's sort of like, you know, the what of translation. Now let's talk about the mechanics of how the cells actually do that. I would say we break it down to like 5 steps. Okay. I'm gonna write here, draw there. Okay. So translation. Step 1. Messenger RNA binds to a small subunit of the ribosome. First, transfer RNA with complementary anticodon to the first messenger RNA codon binds to the messenger RNA. So this is like kind of like A, B, let's see, large subunit of the ribosome joins. So let's draw some pictures. So messenger RNA binds the small subunit of the ribosome. So I'm going to label this as step 1. I'm going to put the small subunit of the ribosome there. I'm going to put the message. I think I will make it purple. Make it something brighter so you guys can maybe see it. Make it blue, maybe. So the message 5' binds the small subunit. We know the message. We're going to use this one. Right, we know that this is AUG, CCU, AGA, etc., etc., etc. Right. So that's A. Message binds a small subunit of the ribosome. First transfer RNA, make those purple. First transfer RNA with complementary anticodon to the message binds to the message. So that's a little more complicated. So I'm going to try very carefully to draw that transfer RNA pretty distinctly. This is the 5' end of the transfer RNA, and attached to its 3' end is an amino acid. We're going to make it a circle, and I'm going to put MET in that circle because that corresponds to the methionine that transfer RNA picked up in the cytoplasm. The anticodon of the transfer RNA is what 3 bases are here at the bottom of that second loop so that there's a match. It's going to be complementary and anti-parallel, blah, 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 right? So what in RNA, what matches with A? U, and what matches with U? A, and what matches with G? C. That's the anticodon. The anticodon is on that loop of the transfer RNA that allows it to bind to the message. We can't read the anticodons in the chart. We can only read the message in the chart. Right? Okay. Messenger RNA binds to most units. First transfer with complementary anticodon to the first messenger RNA codon binds the message. Okay, check. Large subunit of the ribosome joins. So the large subunit of the ribosome is going to come down and bind to this. And the large subunit of the ribosome has 3 spaces in it. Get a little more boldness here. Has 3 spaces in it. We call them E, P, and A. So I'm going to add that addendum on here. Joins with 3 spaces, cavities, grooves, whatever you want to call them, but they literally are physical spaces. This is not their technical names, but I like to remember E is for exit, P is for peptide, and A is for available, or E is for empty, because that's where the transfer RNA is going to go after the amino acid is bound to the next amino acid. So from left to right, they are E, P, and A. Okay, step 2 is easy peasy. Step 2. Next, transfer RNA with complementary, and I'm going to abbreviate now, anticodon to the next messenger RNA codon fills the A site. So this is going to be steps 1 and 2. So my next transfer RNA is going to come floating in here. And it has the correct anticodon, which in this case is going to be GA, right? But it doesn't have methionine on its head. What amino acid does this transfer RNA have attached to it? Proline. I can only draw a triangle, circle, square, so proline is going to be in the square. Fills the A site of the large subunit of the ribosome, right? Step 3. Step 3 gets special all by itself. Peptide bond is formed between amino acid in the P site to the amino acid in the A site. I use my darkish purple for that. So this is important. This methionine is going to be joined chemically, covalent bond to the top of that proline. The carboxyl group from this met is going to join to the amino group of that proline. I'll draw that one separately because that's so important. So step 3. But while I'm doing it, to help you study, right? And this is what I would do if I was you and I was trying to study it. I'm going to repeat all the steps and all the words to help me remember the order, right? So I'm going to draw it again, but while I'm drawing it, I'm going to say it out loud, but you guys can say it like in your heads, right? I'm going to put the small subunit of the ribosome there. My message. 5 prime. I'm just going to put it in AUG, CCU. It doesn't matter. You could make that up, whatever you wanted it to be, right? large subunit of the ribosome joins, right, with its E site, and A site. After that peptide bond is formed, though, this transfer RNA is now empty or uncharged. The second transfer RNA, because the peptide bond was formed, we've got met here. No, sorry. Second transfer RNA, we've got proline here, green, square, pro, right? And on top of that proline, then we have the methionine, because we're making a chain. We're turning individual words in our dictionary into a sentence. I'll make that peptide bond. Step 3 gets its own line. Step 3, peptide bond is formed, because that's hugely important in translation. It's energy expensive to make proteins. It takes 1 ATP to get this assembled, and then it takes 1 ATP each time you bring in another amino acid, and it takes another ATP to charge each transfer RNA. So essentially, it takes about 2 to 3 ATP per amino acid that you add to the chain. All right, so adding that peptide bond, that's a big deal, right? So that's why it gets its own little line. No, I don't want you on right now. Okay, step 4, that's a long one. Lots of words. Ready? Ribosome moves over. It has a fancy name, but we're not going to use it. 1 codon such that colon the empty transfer RNA is in the E site. The transfer RNA with the growing peptide chain is in the P site, P for peptide, P site, and the A site is available. I like that because it begins with A, right? Helps you remember it. Or open, ready to receive the next transfer RNA. Ribosome moves over 1 codon such that the empty transfer RNA is in the E site. The transfer RNA with the growing peptide chain is in the P site, and the A site is available or open. Oh, let's draw that. And while I'm drawing it, I'm going to say it again. So now this is like the 3rd time I've already said it, right? So not just for you to boringly have to listen to me repeat myself, right? But hopefully that's making some neural connection pathways in your noggins so that you'll remember this for the test. So small subunit of the ribosome. Message binds the small subunit of the ribosome. Here's my message. AUG, CU, AGA. But I should make my ribosome a little small subunit. Like the AUG should kind of be not over the bottom of it anymore. Like this, like that. Got my transfer RNAs. Put them in. Right? So, but now my first transfer RNA is over here in the E site. My second transfer RNA that has the growing chain on it. Pro. Very important peptide bond. Net. That one's in the P site, and the A site is open or empty. So I'm going to draw it like this. E, P, A. Right? That was in the A site. Ribosome moves over. 1 codon, chunk. Now everybody shifted positions inside the head, inside the top. The empty transfer RNA is in E. growing peptide chain is in P and A is open and available for the next transfer RNA to come in there, which will have an arginine on its head. Ooh, let's use orange. Triangle. All right. Why is it arginine? Because AGA is arginine in the chart. The anticodon that's complementary to that is going to be, what's complementary to A? This is a transfer RNA. U, C, U. They're good. Step 5. Don't worry, I know you can't see it, but it's not complicated. Go back to step 2 and repeat until a stop codon is in the A site. So I'm going to do this. I'm going to go 5, go back to step 2 and repeat 2, 3, 4, 5. Repeat 2, 3, 4, 5. It's like an endless computer loop until you get to a stop codon. Because the stop codon, there's no transfer RNA that matches the stop codon. So literally the ribosome dissociates, protein gets cut off, protein over. Just as a reminder, I'm going to draw this because I think it's healthy for you to see it and think about it for a minute. What's the one after AGA? CUC, right? So my empty transfer RNA goes back out into the cytoplasm, and this particular transfer RNA that has UAC on it, there's an enzyme that's going to stick another methionine on that transfer RNA's head enzymatically. Okay. The ribosome, right? Now in the E site is going to be this one, right? Because we've added another. My growing polypeptide chain is here, which now doesn't have proline on the bottom anymore. It has my arginine, ARG, proline in the square, and methionine in the circle. I'm gonna get my peptide bonds in there, right? So now P still has the growing polypeptide chain, but now arginine's on the bottom of that chain, and the A site is open or available for the next transfer RNA to fit in there, and then peptide bond is formed, which is going to move the amino acids briefly to the A site, and then the ribosome is going to move over, and that's going to push everybody into the E and the P sites, right? And we just keep doing that until we hit stop. So this one's going to go out and get another met put on it, and then when this one gets kicked out, the cell will stick another proline on that one. In bacteria, of course, this is happening in the cytoplasm. In eukaryotes, technically, it's also happening in the cytoplasm, but mostly attached to endoplasmic reticulum, right? Remember rough ER had the ribosomes on it? That's because in your cells, once this chain gets long enough that it's sticking literally outside of the ribosome, as soon as it gets long enough to stretch over the top of the ribosome, it gets inserted into the endoplasmic reticulum. So that as it's being made, the endoplasmic reticulum can start facilitating secondary structure, alpha helix, beta pleated sheet, maybe adding some things, helping some bonds to form, so that protein starts to take shape rather than just being this chain of peptides, chain of amino acids. Questions, comments, worries, concerns about that? Yes, no, maybe so. No? What time is it? Oh, it's 9:06 already. I want to talk about mutations real fast, so we don't have as much to do on Thursday. I'm probably saying we would have the test, obviously not this Thursday, but probably next Thursday. Is that what we said before? No, we didn't talk about the test before. There's a test scheduled for 2 days from now. That's supposed to be chapters 8, 9, and 6. So hopefully you've been studying 8 and 9 as we go along, because, you know, it's a lot of stuff. 9 will take me one more day. And then 6 is one day. Just a general discussion of viruses. And then it's time for a test. So Thursday, Tuesday, and then it's about time for test. Yeah, there's a lot of connect that goes with this too. Hopefully you're doing it. I did fix number 4, part 2 yesterday. So it should be all right now. Those of you that weren't finished with it, please check back in and see if you can do it now. I think I just forgot to hit save after I gave everybody extension, like I put all the students in to give the extension, and then I forgot to, and I changed the date, and then I forgot to hit save. So it defaulted back to whatever that was, the 10th, the 13th or something. So you should be able to get in there now, but there are other ones accumulating for chapters 8, chapter 6, and chapter 9. And I made a lot of them because I think you need to practice this in order to remember it, right? So there's a lot of connect to do. Do some of it now, some of it later. I think the due dates are all set for this coming Sunday. So Saturday, 27th. Yeah, so you got to kind of get on that, which also I'm going to leave them there for now because I'm trying to force you to think ahead as much as you don't want to, right? And study ahead. Yeah. Okay, so let's talk about mutations real fast, and then it'll give us less to do on Thursday, which then will make you happy because it's closer to the end of the week, right? Get that all copied down. Did you take any pictures? Get me out of the way, whatever. Good, yes, no. Yes, no, I'm gonna erase.
I'm gonna take a picture of it.
I know, we're getting to the middle. This is the hard part. I can promise that this test is the last one that has a lot of stuff in it from BIO1. And it gets a little easier because it's all new stuff. All right, can I erase it now? Yeah. Okay. All right, so then let's talk real briefly about mutations, and then we'll go to the laboratory and cook up some stuff. You're going to be excited because remember the dishes that we put back in the incubator because they weren't finished? We didn't have any micrococcus on them. They all have yellow dots on them today. I checked this morning. This is in your book. It's at the very last of the chapter. Well, we're going to do it now because it's a short piece. And then, like I said, we won't have as much to do on Thursday. So mutation is defined as any change to the DNA, to the nucleic acid sequence. It's plain and simple as that. Any change to the DNA. Nucleotides, let's say. it is one base change, we refer to that as a point mutation. Makes sense, yeah? Because it's one place, one base change, one point. The consequences of a base change, there's many possibilities. One, nothing. We call that a silent mutation. And I'm going to write this one all the way out with all the words, and then the others you'll be able to sub in the right words, right? A silent mutation is a base change in the DNA, which is going to lead to a base change in an RNA codon. Right? However, the RNA codon codes for the same original amino acid. So think about our proline example, which is probably why I pointed it out to you. CC anything codes for proline. So let's say the original DNA strand said CCG. And there was a base change and it got changed to CCA. Well, I guess the DNA would be G's, right? Right? That gives me proline. This C is changed to, probably be changed to a T. Right? The DNA would then say GGT. The RNA would then say CCA. But CC anything codes for proline. So it's silent. Nothing happened. All right. Another consequence can be what we call a missense mutation. Base change in the DNA gives a base change in the messenger RNA codon, and it changes the amino acid that's coded for. So base change in the DNA leads to a base change in the messenger RNA codon. Different. It's a D, sorry. Different amino acid than original codon. Okay. Is that going to affect the protein? Maybe, maybe not. If it's a base change to an amino acid that has similar chemistry, no. If it's a negatively charged base or a polar base, and it gets changed to another polar amino acid, no, the protein will probably still fold about the same and you won't get a big impact. If it's hemoglobin, yes, right? Position 6 has to be valine. If it gets changed to glutamic acid, boom, you have sickle trait. Okay? So sometimes this missense is a big deal, sometimes it's not. Third, nonsense. I'm tired. Nonsense, base change, and I'm just going to use the chemistry triangle symbol for change because I'm tired of writing the word change. Base change in the DNA gives us a base change in the RNA codon, the messenger RNA codon, and the change is to a stop codon. Oops. That will have a big impact on the protein. Because if the protein is, let's say the message is 1200 nucleotides long, and we just truncated it to 200 nucleotides long, that's not going to be the same protein, right? Because the protein's going to literally be like aborted in its process of being made. Now, if different type of mutation, there has been an insertion or a deletion of one or more bases, so we don't really call it a point mutation anymore. That gets a new name. That we call a frame shift mutation. Oh no, I wish I hadn't erased my code over there. All right, so I can still kind of see on the board. AUG, CCU, AGA, CUC, AAA. So that's some of what we had before. Okay. So back to the DNA that was complementary to this. TAC, GGA, TC, T, GA, G, T, yeah, blah, blah, blah. So if I stick another base in, and I'm going to actually put it in between these two G's because it often happens where there are multiples of the same letter in a row. So I'm going to stick, let's say another G gets accidentally put in there by DNA polymerase because it's going so fast, it makes a mistake, and there are G's around it, so it misses the proofreading. Right? Do you see how that would change the message? Everything after that's affected because now if I put another G in there, that's going to code for another C, and now I would have CCC instead of CCU, which I know would still give us proline, but the point is that's going to shift everybody over. So the next base, the next set is now going to be UAG, which is actually a stop. Right, and then GAC instead of, it shifts the reading frame. That's why we call it a frame shift mutation. Reading frame for the sets of codons, the bases that make up the codons are all altered after the insertion or deletion. This would give frame shift mutation. This is usually a lot more detrimental to the organism. Remember, spontaneous mutations occur about 1 in a million bases. Which roughly translates to about 1 in 1,000 genes. Anything physical or chemical that increases the spontaneous mutation rate, we call a mutagen. Mutagens increase the spontaneous mutation rate. And like I said, they can be physical. That means X-rays, gamma rays. I just like to say that. UV light. UV light, that's a more common physical mutagen. X-rays, gamma rays. I don't even really know what a gamma ray is, but like I said, it sounds cool. Okay. And then chemical mutations or chemical mutagens obviously are chemicals that increase the spontaneous mutation rate. Agent Orange, some mushrooms, some toxins, blah, blah, blah. Okay. Free radicals. All right, enough for today. That's enough. That's all.